All right, so the first thing is when we're dealing with this problem set, you'll notice that it says we're going to investigate these cell diagrams. And that the cell is either permeable, we're going to put the cell in a beaker and the membrane is permeable or not permeable to whatever this solute is. We'll see what I mean by that in a second. And there's going to be some substances dissolved in both inside the cell and outside the cell. And you're going to draw a blue arrow to show the movement of water, in which direction water moves. And draw a black arrow to show the movement of anything else that's happening. Answer each question based on the diagram that's provided. The membrane is permeable. This membrane, and all of the problems we're about to go through, the membrane is permeable to glucose, potassium ion, sodium ion, water, iodine, substance X, and substance Z. The membrane is not impermeable to starch and this substance Y. And by the way, when iodine and starch mix, it changes the color of iodine from yellow to black or blue. Now, before we start doing these questions, I got to tell you a few things. Number one, I already told you what assignments are due on Thursday. I will post the assignments in Jupiter grades. I expect that on Thursday you will have uploaded the notes on the video. If you cannot see the video, you need to watch a YouTube video on Osmosis. The notes have to be uploaded. 2, you have the two assignments I've given you. I've given it to you in the ebook. I've also printed them. They're here on your way out the door. If you're missing them, please take them. They're waiting there. Don't get up right now. Please sit down. Not right now. Write your question down. 3, I have another assignment that I've handed you. Could you hold that assignment up and continue? Not the ones from the ebook, the other one. No, not that one, the other one. No?
I got it.
Nope.
Not that.
Nope. All right.
This one does this.
No. That's it. Hold it up. You see that one? That is due, that assignment, which is on diffusion and osmosis and cellular transport, that is due. I gave it to you. If you don't have it again, take it before you leave. Please don't disrupt the class. It's a packet you should be working on Thursday and on Friday and on Saturday and on Sunday. You have all weekend to do it. That is due on Monday. Due on Monday. So these two assignments are due on Thursday. Honestly, you should be done with these already, right? This is due on Thursday. Again, you should be done with this already. The video was a minute and a few seconds long. So they should be done with the video notes. You should be done with the assignment note, the two assignments. Easy, no issues, should be done on already and due on Thursday. I'll have the assignment ready for you to upload. This last one that I just showed you that you've gotten already, a week ago, that I told you wait until after we're finished going over how to do it, that will be due on Monday. You'll have the weekend to work on it. I just gave you a practice test. Everybody got one, right? I'm not collecting that. I'm not grading that. I don't even care if you don't want to do it. If you don't want to do it on your way out, put that practice test back in the pile. I know some kids want a couple copies of this because they want to practice it. That's the word practice test. I will give you the answers, send you the answers to this practice test. So then my concept is that in a practice test, you're going to take it home, you're going to try to do the problems, and as you try to answer them, eventually you'll get the answers and you'll compare your answers to the answers that I send you. If you see a conflict, if you don't understand, you're like, well, number 5, you say the answer is this, but I got this. How? I don't understand. When could you ask me if the test is on Tuesday? 5. The test is on Tuesday. Then you need to ask me questions on Monday over Jupiter grades or during advisory on Monday or during class on Monday or after school on Monday or make an appointment. I say again, make an appointment. Do not just drop by in the morning. And some people keep saying to me, I dropped by and you weren't there. Yeah, read the syllabus. What'd I say? In the morning, I cannot guarantee I will be here. I told you that a million times. So make sure that in the morning, if you're going to come in the morning, you tell me, and I can meet you in the morning, whatever. I don't care. As long as if you have questions, you answer it. Get them answered. And that's it. That's all that's going to happen. Now, we do have a lab. There's a 4th assignment. That's that lab, right? That if you read your ebook, it says such and such lab. That was scheduled for Wednesday, but based on the other classes, we weren't able to finish the practice problems. And so therefore we couldn't get on the active transport. So we're going to spend that time tomorrow, Wednesday, going over that. And we'll do the lab next week, Wednesday. Is that clear? Yes. So Monday's review, Tuesday's a test, lab on Wednesday. Done with that. Let's go on with the practice problems. Please try to remember that these are all permeable. This membrane, and you have to think of it as a phospholipid bilayer, is permeable to these, which means it allows it through, and impermeable to the others. Room 374, Mr. Mendoza speaking. May I help you? Yes. Mr. Williams, go to the office to go home, please. This will be posted. The previous video is posted already. So the membrane is impermeable to what? To starch and why? All the rest of these, it is permeable to iodine, to water, to sodium ion, to potassium ion, to glucose, to X and Z. These can pass. Y and starch cannot pass. And by the way, when iodine mixes with starch, it turns black. It goes from yellow to black, but only when it mixes with starch. Which means that iodine is what we call an indicator. Write it down. Iodine is an indicator. What does it mean to be an indicator? Please answer at your leisure.
To show something, to show something.
It shows what's happening.
That's what I meant to say.
Or what's there. A pH indicator tells you what the pH of the liquid is. That's easy. We're not going to have earbuds in our ears while I'm instructing. Our earbuds are going to be out of our ears while I'm instructing. So indicators show us what is happening or what's there. So if the liquid changes to black when I put iodine in it, what is in there?
Starch.
Starch, because it's a starch indicator. That's what iodine is. Okay, let's move on. These questions are questions you have to ask if you're going to get the questions right on the test. So let me be very, very clear and concise. Your end of course exam, it is guaranteed that you're going to get osmosis and diffusion questions on that test. That will be on your end of course exam. You will probably get them on your Procore exam. You certainly will get them on my tests. When you take AP Biology, you're going to see osmosis and diffusion again. When you take the ACT, you're going to probably see some diffusion and or osmosis questions there. If you take biology in college, which means any science topic that has to do with life, including nursing, veterinary medicine, doctors, anything that has to do with science, with biology, living sciences, you're going to see osmosis and diffusion. If you become a chemist, you're going to see osmosis and diffusion. Are we clear?
Yes.
This is a fundamental process. You should understand that this osmosis and diffusion happens all the time. Diffusion is how you get oxygen into your blood and carbon dioxide out of your blood. Has anybody in here eaten any salty foods, like a bunch of Lay's potato chips or those sunflower seeds are really super salty? What happens to the inside of the mouth, your cheek and your mouth? They feel weird, and the cells, what happens to the cells inside your cheek? It breaks the skin down. What it is that they're shriveling up. By the end of this, you'll see why. When you get in a bathtub and you're in the water too long, what happens to your fingers? They shrivel up. Right? There's a reason for that. Let's take a look at this. Let's take a look at why. So, let's answer these questions one at a time and see what we get. Is the membrane permeable to glucose? Yes or no? Yes, it is. How do you know it is? Because they told you. They have to tell you or show you. You cannot assume that something passes through a membrane. They have to tell you. It's not, they can give you hints. This substance is nonpolar. If it's nonpolar, it's going to pass through. If it's polar or charged, you have to have a carrier, right? A transporter. You have to have a protein, because polar or charged particles need something to let them through. Otherwise, they're not going to get through easily. Non-polar small objects particles will get through easily. So oxygen gas does not need a transporter. All it needs is it'll pass right through the membrane. Yeah? Yes, you may. So glucose will pass through. So yes. In what direction will glucose go? Into the cell or out of the cell? How do things move? What's the easiest rule in all of science? How do things move? From high to low. The easiest rule in all of science. If you can't get this, I don't know what else to tell you. Things move from high to low. So where is this high? 90% is pretty high. What's low? 10%. In order to tell which side is high, what do you have to know? Which side is low? And you have to decide, how do you know what is, when you say side, how do you know what side it is? We're talking about a cell. You're talking about a cell in a solution. So the solution is one side, and inside the cell is another. So you're talking about inside versus outside. Inside versus outside. Are we clear? Two different conditions. All right. So water is going to go from, or glucose is going to go from the outside to the inside. Now, what is the percent water on the inside of the cell? 90% water. How do I know that? Because there was 10% glucose. There's 10% glucose inside, that means there's 90% water, percent. The total has to equal 100. So if it's 10% glucose, it's 90% water. On the outside of the cell, if it's 90% glucose, it must be 10% water. All right, let's move on. What's the percent water inside the cell? It's 90%. What's the percent water outside the cell? It's 10%. Will osmosis occur? No, because if the solute can move, the water won't move. Am I clear? If the solute's going to move, the solute's going to equilibrate. It'll get to equal, and the water won't move at all. So you don't have to worry. So no, water osmosis will not occur. There will be some movement of water, but in general, the glucose will find an equal state. And remember, what does that mean? That means that this 90 is going to come down, right? As glucose leaves outside, it goes inside. The 90% goes to 80%, goes to 70%, right? And the 10% is going to go up, right? From 10 to 20 to 30 until they get about equal. Do you agree?
Yes.
So it's going to be about 45% inside and outside. Right? It'll be about 45%. It has to be somewhere between 10 and 90, and the midpoint is going to be about 45%, right? Once you get to 45, it stops moving. We're good. The movement of water will cause changes in the shape and size. Will this cell shrink or swell? Neither, right? Neither. Why won't it move? Why won't it shrink or why won't it do either? Water will not move. Because why? The glucose moved. The solute will. The solute will move and everything will settle out. Are we clear? We're going to let one more person go. Let it be the one that has the most need, because I'm not going to have a revolving door in this class. So one person choose to go. You guys figure it out. I got a class to teach, so you guys figure it out. Is the solution in the beaker hypotonic, hypertonic, or isotonic? Well, to answer that, we really have to understand what it means to be hypotonic, hypertonic, and isotonic. So let's draw three different situations out. One of them is going to be hypo, and the word is tonic. Move if you can't see. And the other one is going to be hyper, again, tonic. And the last one's going to be iso, and we're going to say tonic. Very good. Higher than what? Higher than the solution, right? So that's good. Somebody already figured out the hardest part. Hyper, by the way, the word tonic means the solution. All right? So hyper means higher. Somebody figured that out already. They know because we've used the word hyper in our society all the time. So hyper, outside, higher outside. So let's say it's 10 inside. What's it going to be outside?
90.
Or what? No, don't say 90 because you're trying to add it up to 100, aren't you? We're not gonna do that because it's 10% solute inside. So what is it? Water inside. 90% water. So let's erase this. And you guys don't confuse this. The two situations are separate. They're different. Outside and inside are different. So if it's 10% solute inside, then it's 90% water inside. Okay, that's true. But if this is hypertonic, someone said it means it's higher concentration of solute outside. That's what hypertonic means. What do you know about the outside concentration of solute? Higher than what? Than the inside, which is what? 10. So it has to be higher than 10, right? In this case. So 10.5. 10.1. Whatever. 50. 100. Whatever it is. Higher than 10. Right? And if it's 10.5% solute, what is it? Water.
Oh, wait, 89.5.
That's right, 99.5% water. Does that make sense? That's just one example. If I would have put 5% inside, it had to be higher than 5%. This could have been 90. It could have been 90.7429. It could have been whatever, right? But as long as it's higher than what's inside. That's all that hyper means. That's it. Hypertonic. Now hypotonic, what do you think that means?
That's less.
Lower concentration. So now if I have this cell and I have 10% inside, which means what's the percent water? 90% water. What must it be outside? Less than 10.
Yeah.
So it could be 9.9% if you want. It could be 8%, it could be 2%, anything less than 10. And what is it water then if it's 9.9%?
80.1 or 90.1. Smart.
90.1% H2O. So whatever it is, hypotonic means it's less than what's less, the solution is less than what's inside the cell. Hypertonic means it's higher than what's in the cell. What about ISO? Equal to. Excellent. That's what ISO means. ISO means equal.
Wait, for the hypotonic, you said that's 90.1%? I thought it was below. That's 90%.
Yes, that's true, but it's the solute that has to be higher.
Okay.
The solute that has to be lower. The solute, that's very good. Somebody noticed a very important point. When we're saying hypertonic, hypotonic, we're saying the solute concentration is higher. In the case of hyper, higher than this inside of the cell. If we're saying hypotonic, the solute concentration is lower than what's inside the cell. And ISO, of course, means it's the same. They're the same. And of course, when you have ISO, there's no movement. You should note that when you have a higher concentration of solute outside, if water's going to move out. You should make a note of that because we're going to talk about that in just a second. If you have a situation where the solute concentration is higher outside, you have a hypertonic solution, then water's going to move out. If the solute cannot move. In a hypotonic solution, water's going to move in. If what? If it's lower. If the solute doesn't move, then water's going to move in. We'll talk about why in a minute. Just remember that rule and we'll continue. Are we all good with this then? Go to another example problem?
Yep.
I think we only have one problem on this. All right, so that's the first page. Let's go to the second problem. It can get really crazy. All right, so let's take a look at this one. I'm going to let you answer these on your own. Go ahead and answer them on your own. The next, maybe I'll have to do a couple as a group, but right now, go ahead and try to answer on your own. I'll give you 3 and 4 minutes. Do we have this yet? Or yes, of course. That's what I want to do.
Why do you also talk?
This is why teachers try to hit her in cars. All right. All right, let's get through this. All right, glucose, will glucose diffuse?
Yes.
How do you know glucose will diffuse? Because it's permeable, good. Yes, the answer is yes. In what direction will it go?
Into the cell.
Into the cell. Does anybody not understand that? Please say that now. If you chose something not this, why? Yeah, go ahead.
I thought since it was 10% glucose, it would go outside the cell to the 60%.
Okay, so you have it reversed. So let's think about this, and please try to remember this. Get in your head, guys, everyone. Diffusion, this is the word diffuse, right? Diffusion is what? Diffusion is from high to low. High to low. You got to get that in your head. Saying it for 2 days now. I know some of you weren't here yesterday, but you got to put in your head, high to low. 60 is high, 10 is low. Well, what about the water? What about the starch, Mr. Mendoza? Does the starch move?
No.
No, it does not move. Stays the same, so we're not going to worry about that. Now, here's a question. If the starch does not move, glucose is going to move until it's equal, right? But will water move now? Ah. Now things get tricky. Oops. Right? Because, okay, the glucose will move until it's about, what, 50-50 or, whatever the percentages are going to be on both sides. So let's first think about what's the percent water outside. If we got 60% glucose outside, 10% starch outside, what's left?
30.
It's 30% water outside. But the glucose is going to move until it's equal, right? So what if we just ignore glucose then? And we just said starch. The glucose will be equal at some point. Then of the leftover water, right? Of the leftover solution. Can you work with me here and think about that? Do you want to make up a number?
No.
You think this will be 35 glucose outside and 35 inside? All right. So let's say that. Let's say it's 35% glucose outside, 35. When it's done, right? Unfortunately, this is all happening at once. You can't really separate it. But let's say that happens. What's the percent water outside then?
On the outside.
35 and 10 is what?
45.
45. So what is the outside percent? 55% water outside. Agreed?
And 5% inside.
And you got 60% starch inside and 35. 60 and 35 is 95%. So it's about 5% water inside. Do you agree?
I didn't get that.
What did you not understand?
I just added the glucose and starch together and then used the remaining amount.
Which you can do. You can do that. I'm just trying to get a hold of either way you're going to get the same answer. Okay? If we got rid of this is all best guess, right? But let's say we don't do that. What if we just say, look, what if we just stick to the original numbers? Now, by the way, water was there's a much higher water concentration outside than inside in our last discussion, right?
Yeah.
So Yeah, unfortunately, you have to think about that. You have to think about the fact that this is gonna get less glucose and the other's gonna get more glucose. Do you understand that?
Yes.
The glucose is gonna move, and no matter what you say, whether it's 35 or 40 or whatever, right? The bottom line is that this is gonna get less and this is gonna get more. Do you agree? Do you see that? Let's start with that. Do you see that glucose is gonna diffuse? Yes or no?
Yes.
If you're staring off into space, I need to know that you're responding to me, that you understand what I'm saying. I don't want to say your names, but you weren't looking at me, so I'm not sure, right? Do you understand that glucose can pass by the membrane so that this glucose is gonna rush in and they're gonna even out to some number? That number is gonna be higher than 10 and lower than 60. Do you agree? I don't mind putting 35%. The number to me is not that important, but let's say it's 35. What we do know is the two percentages of glucose inside and out are going to be what? Eventually, going to be what? Even. They're going to be equal, right? So glucose will keep moving. Let's say 35 and 35. Let's just say that. Is everybody okay with that?
Yes.
So 35 plus 10 is what? 45. That leaves how much water?
55%.
55% water. Inside it's 60% and 35%. What is it? 95%.
5% water.
I'm sorry, 5% water is what's left. Do you agree? So we know water's going to move. How do we know water's going to move? Hold on to your question. I want to answer it, but how do we know water's going to move? Yeah.
Because it's more on the outside than there is on the inside.
Because it's higher outside than it is inside, and it can move. It's permeable to the water, because sometimes membranes are not permeable to water. A Ziploc bag is a membrane. It's not permeable to water. It's watertight. Well, it's 8:57. We're not done with this, so we're going to have to finish this tomorrow, which means we can't do the lab tomorrow. We'll have to do it next week, but we'll get this lab done. So we know that starch will not diffuse. Let's go ahead and write that down. No, it will not diffuse. And how do we know starch will not diffuse? They told us. They have to tell you if something diffuses or not. You cannot assume. The membrane every membrane that works differently. So there's no way to tell unless you see something happening in the problem. If I show you stuff moving across the membrane, then you know it's permeable, right? In some way, they have to tell you if it's permeable or not. Is that clear? And they clearly stated that this starch was not permeable. So starch will not be moving. So what does that mean? So go ahead, what? Exactly. That means the water's going to move. So there's two ways to do this now. In which way is the water going to move? There's two questions you have to ask. Okay, there's two ways to do it. I'm going to do them both ways. You ready? Well, the starch would move out, but it can't. So water's going to move in. That's one way. The second way is, well, water's higher concentration outside than inside, so water's going to move in. In both ways, where's water moving? In. So the water's going to move in. Are we all in agreement here? Is there any issues here? Water's going to move in. What's that going to do to the cell as the water moves in? It's going to expand the cell. It could blow it up, unless it's a plant cell. Blow it up, I mean, it could rupture the cell membrane. What is the percent water inside the cell? At this time, the original portion is what? It was 5%. Now we know at equilibrium it's going to be higher, right? The percent water outside that was 55% and will be lower, right? Will osmosis occur? Yes. If so, in what direction? Into the cell. Yes, you may. Is the solution in the beaker hypotonic, hypertonic, or isotonic? Initially, the initial solution. It used to be what? 70% solute, right? 30% water outside initially. Do you agree?
Yeah.
Let me erase this so we can look at the initial. I'm going to erase this because we did all the work already.
It was 70, so it's isotonic.
So, that's right. It was 70 outside and 70 inside. So what was it initially? Except that it wasn't, was it?
Oh, yeah.
Because the starch, for glucose, I mean, it was 70 and 70, you're right, in the beginning. But the problem is starch can't move. So you have to look at 60 and 10.
Oh, so it's not.
Do you see what I'm saying? Focus on the fact that this can't move. The glucose will equilibrate, so you don't have to worry about that. So you can ignore glucose, but the starch won't move, so that means it's going to be 60 out inside and 10% outside. Is that clear? So realistically, it's even though you could argue it's an isotonic solution, the argument would be false because because starch can't move. It's actually a what? Hypotonic solution. And that explains why water can move into the cell. Is that clear? A very difficult question, really, right? Because it's very tricky. You see how tricky everything was? If you just went by the general percentages, you'd say it was isotonic. But you have to think about, this isn't moving. It will never move. The glucose will move and equilibrate. So I can ignore the glucose for the purpose of discovering whether this is hypotonic or hypertonic, because they'll both be equal. So the only question then is this starch. Starch is 60% inside and 10% outside, so obviously this is hypotonic. By the way, what does that tell you? Every time it's hypotonic, where's the water going to move?
Inside.
Into the cell. Every time it's hypertonic, where's the water going to move?
Outside.
You see? In ISO, the water won't move at all. Yeah, question? After someone comes back. You saw someone left, didn't they?
Yeah.
Am I right?
Yeah.
Okay. Sometimes I'm trying to focus on what I'm doing. And try to do this on your own. Let's just review real quick because you may not remember. Does water move?
Yeah.
Yes, it does. This is in distilled water. Quickly, does potassium move?
Yes.
Does sodium move?
Yes.
Does starch move?
No.
Is this hypo, right now, as it stands, is this hyper, hypo, or iso?
Hyper. It's hyper, because it's 100%.
It's 100% water. There's no solute in here. This is about as hypo as it gets. You can't get more hypo than zero, right? I can't imagine having a negative because what is it, a black hole or something? It can't have a negative number. So the concentration of solute is zero here, right? So it's 100% water. So that's as hypo as it gets. All right, so you guys can answer the rest of these. I'll give you 5 minutes and then we'll get back. We'll go walk through the problem set. The problem. I'd like to see you writing it down. Write down. Draw it. Write down the answers. Just write them down. What? Oh, it's recording. Sorry. I'll stop. So water will move into the cell. Why? I just said it and I did not record it, so I'll say it again. 1, it's 100% outside, 70% inside, water will move in. 2, if starch can't move it out, water will move in the opposite direction, into the cell. 3, it's hypotonic. Hypotonic means water moves into the cell. Will potassium move? Yes, it'll move out from 10% to 0. Will sodium move? Yes, it moves out from 10% to 0. How do I know they're permeable? They told me at the beginning of the problem set. What's the water concentration inside the cell? 70%. How do I know that? 30. 100 minus 30 is 70. Concentration of water outside is 100%, obviously. Will osmosis occur? Yes. Into the cell. Will the cell swell or shrink? Swell. Why? Water's moving in. I hope so, because it's not that hard as long as you guys break it down into those simple questions. You got to ask those questions. If you miss even one question, you're going to mess it up. Yeah, life is confusing. Is the solution of the beaker hypotonic, hypertonic, or isotonic? It's hypotonic. It's as hypotonic as it gets. You see 100% outside, and anything other than 100% inside, the only thing you can, if you see 100% water outside, the best you can do is isotonic, right? You can't get lower than 100% water as far as solute concentration. All right, so that's the next two questions. So here you have hyper. That's what we say now, right? Hyper. So he says hypertonic. Why? Because 20, 20, and 10, right? And that leaves you 50 and 70. But now consider what's going to happen when the glucose moves in. What are you going to end up with at the end? The 20 is going to go to what? 10% and 10% inside.
Wait, how?
Because it's evening out.
Because it's going to keep going until they're even. If it's 20 outside, it's going to keep going in until what? Until both sides are equal. And it's 20 outside, what's it going to be inside? And what's half of 20, man?
10, but it's
10.
only 5% of glucose inside.
So it's going to...
Or take the go in.
Yeah, that's what he's saying. It won't be even if you add 5. It's gonna be.
Oh, I see what you're saying. All right, so let's say... Well, I don't know. Just make up a number until it gets...
12.5.
Whatever. I just say 15. All right, 15%, 15%. The numbers is not the point, right? There's gonna be some number. Is that clear? So 15. We could figure out the exact number. I don't want to waste our time. We don't have a lot of time.
I know I'm saying for a test, will it matter?
No.
Okay.
All right. So 15. The only real reason they give you these problems is to figure out which way directions the water are going to flow. Is the ion going to flow in which directions they're going to flow? They want to understand, do you understand how things move across the membrane? That's it. So 15 and 15. So potassium is 10 outside. What's it going to be inside in the end?
10.
10 and 10. That's easy, right? And iodine's going to be what?
5.
5 and 5. Everybody okay with that? So now, but look at what happens. Outside, the total solute now, the total solute outside is what?
30.
So we started out hyper. The total solute's 30. It's 15 plus 10 plus 5, right? So that's 30% solute. What does that mean for water? 70% water. And inside,
55%
inside we have 5, 15, 10, 25, and 5, which is what?
55%.
55% solute. Right? So what is it? What's the water inside now? What is it? 45%. So now where's the, what is this now? What is the solution now after the ions have moved?
Hypo.
It's hypo. So in which direction is water going to go?
Inside the cell.
Inside the cell. Do you see the change? Do you see why it depends on when you're talking about the movement of water, when are you talking about the movement of ions? It all depends, right? So anyways, it's hypotonic. So now if we asked, will osmosis occur? Sure, it's going to go into the cell. I always say do the ones that do the ions that transfer first. Don't worry about it. Will any other substance diffuse across the membrane? Yeah, everything is going to, everything except starch. That's glucose. That's potassium ion. That's iodine. Will go. What direction will each substance diffuse into the cell? Except for water, of course. Actually, water will go into it as well. Is the solution in the beaker hypertonic or isotonic? Again, depending when, but yeah, hypo at this point, right?
So when we get it wrong, if you put hyper, since it was hyper.
It will be much clearer on your test.
Okay.
All right, these are practice problems. They're meant to challenge you, to make you think, right? What will be the initial color of the solution immediately after the cell is put in the beaker? The outside's going to be, immediately, the outside's going to be yellow, right? Because what color is iodine? They tell you it's yellow. In the solution, right? In the cell, I'm sorry, that's not true. In the cell, it's going to be clear initially, right? And the beaker is going to be yellow. Because iodine's yellow color. But then after the diffusion, see how there's a difference between before all the diffusion and osmosis and then after, there's a change. We have a drawing, but the drawing isn't showing you what you have. It only shows you where you begin.
Could you do
Right.
it live?
Huh?
Could you do it live with this?
We are doing a lab. We were scheduled to do it today, but do I need to go through this again? All right, thank you. So the cell, after the fusion, what color is the inside of the cell? Bluish black. Why is it bluish black?
Because starch is mixing with iodine.
Because that's right, because as soon as starch meets up with iodine, it turns bluish black. That's why that iodine is an indicator of what? The presence of what? Starch. So it's one of our indicators in biology. It's an important indicator for biochemical processes. So what color then is the outside? We already said it's still going to be yellow. The outside is still going to be yellow. Why is the outside still yellow if the inside is...
Starch didn't go out because it can't go through.
That's right. Starch did not leave because starch is impermeable. So it might have turned a little less yellow. It might have gotten a little paler because you have less of it out there, I guess. But that's about it. All there would be to it, right? Are we all good with this then? All right. Now, the final color, I think I said down here, the final color inside the cell is bluish black. And the beaker is going to be yellow because there's no starch coming out, assuming that everything works the way it's supposed to. And yes, we will do this lab. Let's go back and do this one up here, number 4. Will osmosis occur in this situation? Let's take a look. Yeah, we know it's going to occur. Why is it going to occur?
Because there's starch.
Because you got starch underneath here, and that's not going to move. And there's no starch outside. So we know there's going to be some osmosis going on. I always say let's do the ions first that can move. If you have 40% outside, 38% inside, what direction starch is glucose going to move? Into the cell. And you're going to get probably what, 39, 39?
Yes.
You're going to go, you're okay with that? And sodium's going to do what?
Go inside the cell.
Into the cell. And it's going to be what? 18, 18?
Yes.
I mean, the exact numbers for our purposes are not that important. What it matters is that you understand that it comes to some kind of equilibrium. And potassium's going to do what?
Go outside.
It's going to go out. Very good, you got this. Potassium's going to go out. It's probably going to be about 2, something like that. All right, so outside you have how many? 20 plus 39 is 59% solute outside. Inside you have what? 64% inside. So 59% solute means what percent water outside? 41% water outside. And inside the cell you have what percent water? 37% inside.
Wait, no,
36.
36, I'm sorry.
36% inside. All right? So in what direction will water flow? Because we said osmosis was going to occur
Outside.
into the cell. There's three ways to think about it. Let's go through all three ways one more time. So the first way is starch is not permeable, but starch would go out if it could, all right? But it can't. So what? Water will go in. I don't know why people are not finishing my sentences. So because glucose starch would go out if it could, but it can't. So what's water going to do? It's going to go in to make sure until starch is equal with it, right? So that's one. That's one way of thinking of it. Another way of thinking about it is since you have 41%, since water is at a higher concentration, I'm going to put square brackets for concentration, outside, then water will move from out to in. So that's another way of thinking about it, right? How did I figure that out? Look, it's 41% outside, it's 36% inside, so water's going to move in, right? Then the real quick way, if you can remember, it's probably the easiest way, the quick way to do it, is outside is hypotonic. How do I know it's hypotonic? Because it's high concentration of water, which means it's a what? If it's high concentration of water, what is it? Low concentration of solute. So hypotonic means water moves in. Is that clear? That's it. That's all there is to it. Yeah?
So, say if the cell was like hypertonic inside, then that means that it has a low amount of solute.
That's exactly what's happening. Hypertonic. The cell is hypertonic because it has a higher percent solute, but it has a what? Water. Higher solute means what?
Lower concentration.
Low water. High solute means low water. High water means low solute. Yeah?
So even if we diffuse them evenly, we still gonna get the same thing? Like,
If what now?
even if we don't diffuse it equally, we still gonna see that water goes.
Sometimes it changes the problem. We saw that in problem 5, if we just took our initial analysis was what? The outside was hyper, but then when we looked at it after the diffusion occurred, it was hypo. Without the without osmosis, it was hypo. Over here, we started with 40, what was it we started with? Somebody help me out here. We started with 40, 20, and 10, right? Or no, 40, 20, and 1. And inside it was 38, 15, 3, and 5. So we always started with a higher concentration of solute inside. So in this problem, it would not impact, right, the movement of water. We would always know that water is moving in this problem because it has no choice. In this case, the concentration of solute inside is always higher than that outside. So, and of course, the 100% water problem also, it didn't change that problem either. But you saw that number 5, it changed the problem. So you got to be careful. I can promise you on the test and in the course exam, they're not going to give you these kind of multiple layers. If they do, you won't have to, it won't be like this. This is just made for you to think. All right? So will osmosis occur? Yes. In what direction? Into the cell. Will any other substances diffuse across the membrane? Yes. Which ones? Glucose, sodium ion, potassium ion. What else? That's it. In what direction will the substance diffuse? Glucose and sodium ion go into the cell, and potassium ion goes out of cell. Will the cells shrink or swell? Swell. Are we good with this? You guys feeling comfortable?
Yes.
Is the solution of the beaker hypotonic, hypertonic, or isotonic? Let's say at the end, is it hypotonic, hypertonic, or isotonic? Hypo. Well, after diffusion, but before osmosis, it was hypo. And we're good? And that's how we do it. All right, so let's go on to the... And then we got 2 more problems, but we won't finish them today. We'll finish them tomorrow, then we'll do active transport, passive transport, and I think you'll be ready for your test then. Good luck, clean up, thank you for your time.
Can you go back to?
I don't want to go back. I want to finish these last 2 and start helping you with your homework that you have to work on for the next 3 days. Right? Those are the book problems. They're very straightforward. We went over them. I printed them for you. Am I clear? I'm not going to go over it again. I'm not, because I don't have time. So you have 2 handouts I printed for you and gave them to you. Said these 2 are due on Thursday, along with notes on the video. So those 3 assignments, I listed them out. Go back, look at the video I posted before, and go back and, or maybe it's at the beginning of this video. No matter what, go back and look at it, please. Good Lord, we're going to spend more time on this. Go ahead. Go ahead. I feel that you don't hear the clock ticking. I hear the clock ticking away. Every second means something that's not, you're not going to get help with. Do you understand? All right. Huh?
It's in the beginning.
Wow. Okay. All right. Let me just move on because I can't. I'm going to lose my...
Everything that he said, he wrote it down in the beginning.
So let's go on and look at this. What do we know about X and Y? Or X, which one passes by? Does anybody remember? X and Z go past the membrane, Y does not, right? So Y is going to stay there. Starch does not. So I'm gonna put a little red line underneath the ones that are not going to move. All the others move, right? So that makes my life easy. And glucose is going to move in which X is going to move in which direction? Out. It's going to move out, inside, outside, right? Until it's about 2.5 inside and 2.5 outside. Do you agree? I'm talking percentages here. Z is going to do what? Same. It's going to move out until it's about 2.5 and 2.5. Y is not going to move. Starch is not going to move. How about glucose?
Yes.
It's going to move in to 1.5 and 1.5. See, all those would be if those were the only ones at the end of diffusion, would be isotonic, which is why there would be no osmosis, right? There would be no swelling or shrinking really, because the ions would just move and you'd have
So it's not isotonic because of the Y and the starch.
the Y and the starch. It really if it depends on the question you're being asked. If they put up a cell and they say, is this? They don't ask you which direction things are going to move. Am I clear? Don't get confused about this. If they give you these percentages, they say glucose, starch, X, Y, Z, and they ask you, is the solution out here isotonic, hypertonic, or hypotonic? Then all you do is add these up. That's 15%. This is 5%. What's the solution? Hypotonic. That's it. Am I clear? But if you're given these kind of like, these are going to move, let's work out the problem, let's think about it kind of issues, then you know these are going to move back and forth. So outside you have what percentage? 2.5 plus 1.5 plus 2.0 plus 2.5 is 8.5%, which means what percent water?
91.5.
91.5, oh jeez, I put 9.1. 91.5% water. So now let's do inside. Inside is what?
10%.
Inside is 10%. It's 2.5, 2.5, 2.5. This yet it's actually 11.5%, right? And what's the percent water then?
88.5.
88.5% water. So that's what we have. What's going to happen then?
Osmosis.
Osmosis, water's going to go into the cell. Water's going to move into the cell in order to equilibrate that concentration. All right. Now, what substance shows net movement into the cell? Well, we know that it's glucose, and we know it's not starch, and that's the only one that goes into the cell, besides water, right? What substance shows net movement into the beaker? X and Z. No, we said water's net movements into the cell, isn't it? Didn't we? I'm confused. You're confusing me. All right. Does osmosis occur? Sure. Into, right? Will there, if you were told that one of the molecules X, Y, or Z was the polysaccharide glycogen, which would you guess would be the glycogen and why? Well, it's not, glycogen is not starch. How do you know it's not why?
I think it is why.
Why do you think it's why?
I think it's why because our cells store glycogen, and if it was permeable, then they wouldn't be able to store it right.
That's so genius. It's a very intelligent way of thinking of it, yeah. Yeah, that's very good. Well, here's another thing you should know. What is it that membranes don't allow through the cell? Give me a list of things that membranes don't let pass through easily. Cell membranes. Give me a qualities. Give me some qualities. Things that. Well, we said starch, but think about it. What are the basic qualities of stuff that will not pass through? Yeah. Yeah, but think of more basic. Some large particles. So anything big, right, is going to be a problem. That's why a bacteria won't get through easily because it's big, right? Compared to like a protein or compared to an ion, right? So large is no good. So this again, hold on, let's not worry about imaginary because imaginary we don't know what qualities they have. So let's go and say do not pass easily across the membrane, right? Large. What else doesn't pass easily? What needs help? Color is irrelevant. What needs help crossing that membrane? Come on, guys. When you see this line, what are you supposed to be thinking about? What is this made of?
Phospholipids.
Phospholipids. What's in the center?
Hydrophilic.
Hydrophilic's in the center.
No, that's hydrophilic.
Hydrophobic's in the center. That's what you're saying?
No,
That's right. It is hydrophobic is in the center. Come on, guys. Hydrophilic heads, hydrophobic tails. You've got to get this in your head. So you have the hydrophobic center. Does that allow, what does that not allow to get through easily? Hydrophilic stuff. What is hydrophilic stuff? Stuff that's charged or polar, right? Charged or polar does not get through easily. Charged or polar. Now, what do we know? What do you know about starch and about glycogen? What do you know about those? Yeah?
I think glycogen
But-
was how animals.
That's right, how animals store sugar. Very good. So you know it's made of sugar. And you know starch is made of sugar because that's how plants store sugar, right? Okay. I told you about 5 times in this video and in the previous video. You're probably not hearing me because some of you are having conversations instead of listening, but that's okay. Sugar is polar and charged, right? What else do you know about glycogen and starch?
They're,
Is it big or small?
big.
Big, very big. It's a long polymer. It's a very big polymer of many, many glucose molecules. Both of these are large polar molecules. Very large. Chains. Remember the pictures that I showed you? Big, long chains? Go back and look at the video if you don't remember it. Big, long chains of sugar. Is that going to pass through the membrane easily? No. So it's not going to be, this membrane is probably not going to be permeable to starch or glycogen. Which one of these are these not permeable to?
Why?
Y and starch. We know starch is not glycogen because it's starch. So which one's glycogen? Y. You knew that though. If you would have spent time thinking about it, you would know which one it is. If you think about what is starch. What did we do? We thought about the qualities of what starch is. We thought about what the qualities of glycogen were. And then we answered that question. Don't just make guesses. I don't tell you things just because I want you to memorize some ridiculous fact. I tell you things that are important that you will use if you remember them. All right. So it is in fact Y, and because it's big and polar, and therefore not easily permeable. So they actually gave you a clue. Remember what it is to be a polysaccharide, right? What is it to be a polysaccharide? A polymer of sugar, large, polar, charged ends, right? Oxygens everywhere. Oxygens and hydrogens. What do you get when you get oxygen and hydrogen bond? You get hydrogen bonding. What does that mean? These things have polarity. Are we clear? So, and we know it dissolves in water, so we know it's polar, but starch and glycogen don't get through that membrane. Yeah.
So you know how monomers of glucose make up glycogen. So is that saying basically that the glucose that's in there is just a monomer, not polymers?
That's good. The reason they say glucose and then they say starch and then they say glycogen, because glucose is just a monomer of sugar.
It's just one.
Just one. One hexose floating around in water. This starch is a bunch of those glucoses all bonded together. And this glycogen is a bunch of those glucoses all bonded together. Got it? All right. Let's go on and look at number 7. What substance shows the net movement into the cell? Well, let's remember, does Y pass through the cell?
No.
No. The starch?
No.
The starch doesn't. What's the only one that's going to pass by?
Iodine.
Iodine is the only one that's going to pass by. By the way, at the beginning, is this hypertonic, isotonic, hypotonic? Look at it. Outside it's what in the beginning? 15% solute, and inside it's 10%. But then after iodine moves in, and you get, what are you going to get? You're going to get something like, I don't know, 6.5 and 6.5, right? Something like that. So now outside it's what? 8.5, and inside it's what? 16.5. So now you went from a hypertonic to what?
Hypo.
So after diffusion, there's a different answer, right? So after diffusion, starch is, so that's why I tell you, if you want to wonder about water, you really want to worry about what's happening at the end, right? what really, the only thing you want to worry about osmosis, all you worry about is which one of these is not moving. The one that's not permeable is going to determine osmosis. Does that make sense? Because all the other ones are going to equilibrate. So outside, really, I'm going to say, let's not worry about what it was before we did the diffusion. Let's worry about what it is after the diffusion. So it's 8.5 and 16.5. And so we know that, what is the percent water outside? 83.5.
Oh, I was first thinking 3.5. That one's 91.5.
This was 91.5% water. And what is it inside? 83.5% water. So which direction is water going to move?
Into the cell.
Well, how do I know it goes into the cell? Those three rules, right? Those three rules tell me that this is hypotonic. How do I know it's hypotonic? So less solute, more water, right? I know water is bigger concentration outside than is inside. I also know starch won't move. Starch would move out if it could, but it can't, so water's going to move in the opposite direction. Y would move out if it could, but it can't, so water's going to move in the opposite direction. That's it. Are we good? So iodine, what substance showed net movement into the beaker? Well, water. Iodine.
But what into the beaker before you say?
Oh, into the beaker. My bad. My
Then
bad. Into the beaker would mean none.
None.
Does osmosis occur? Yes. Into the cell. At what point does the experiment, what color would the cell be? At the end of the experiment, what color would the cell be?
Bluish black.
Bluish black is correct. And why would it be bluish black?
Starch and iodine.
Because the starch is ionized. And what about outside? What, even at the beginning, what would it be?
Yellow.
It'd be bluish and black.
Because there's iodine and starch out there too. All right. Are these problem examples of active or passive transport? It is passive. Good. You guys have been doing your reading. Good. It is passive. Diffusion is passive, which means the difference between active and passive is active requires energy. If you're going down the concentration gradient, if you're going from high to low, it is passive. Okay? It's going to get a little tricky in a minute, just so you know. All right, let's